Good morning, everyone. First of all, I would like to thank you very much for inviting me to come here. I will present some information about our work in Belarus. I'm involved in invasive alien species monitoring program, and my key taxonomic interests are alien species of invertebrates and indicators of ecological water quality in Belarus and neighboring countries. I was previously involved in CBD GTI training course on rapid identification of invasive alien species for achieving Aichi biodiversity target 9 using DNA barcoding techniques that took place in 2015 at Biodiversity Institute of Ontario, Guelph University. I also was involved in Global Malaise Trap program that was conducted during 2015-2016 using one trap that was set in the Central Botanical Garden of the National Academy of Science of Belarus for 40 weeks. 50,300 sequences of animals and plants from Belarus exist in BOL system. Most of them belong to arthropods, and I need to mention that almost 98% sequences were obtained and uploaded to BOL during CBD GTI program and Global Malaise Trap program. A bit history of DNA barcoding in Belarus. In 2014, Belarus State University joined RUS-BOL as an equal and rights member, thanks to productive and excellent work of Nina Voronova. She is Associate Professor of Zoology Department who works on pest insects, especially aphids. In 2018, 8 local participants of Institute Genetic and Cytology and Scientific and Practical Center for Bioresources and 16 participants from 7 countries like Belarus, Ukraine, Moldova, Georgia, Armenia, Azerbaijan and Kazakhstan were selected for participation in GTI-BBI joint training course on using DNA technologies for identification and study of invasive alien species and endangered species. During the training course was decided to establish regional DNA barcoding network, and 2 institutions of National Academy of Science of Belarus were chosen as lead institution. Different representatives from 4 spheres, research, education, private sector and government were involved in regional DNA barcoding network. All name of organization involved from 7 countries you can see on this slide. At this moment we don't have any funding, but we are going to apply for bilateral and international project. Because of regional network is big and there are different small projects in each country, I will mention the general idea and way of our joint research that correspond to main iBOL aim. First project is library construction, where Belarus is a project lead in collaboration with Ukraine, Armenia, Georgia and Moldova started to create library of aquatic alien species of invertebrates and fish during GTI project, and we have planned to continue. GTI project ends in December 2018, but we hope it will be continued until March 2018. Our goal to check the existing list of alien aquatic invertebrates and fish in Belarus, Ukraine, Armenia, Moldova and Georgia using DNA barcoding. The number of species you can see here. Second project is also about library construction. Together with Ukraine, Armenia, Georgia and Moldova, we are planning to fund financial support to create library of barcodes for Ephemeroptera Plecoptera Trichoptera group. Species of that group are indicators of water quality, and used for water status assessment according to Water Framework Directive. This group is pretty big, and all numbers you can see on this slide. Third project is about meta-barcoding. In this year, together with our colleagues from University of Otago, New Zealand, we applied for the financial support to conduct the project on monitoring of aquatic invertebrates and fish of Belarus by meta-barcoding. Last but not least, Belarus SWOT analysis. In Belarus, we have high morphological expertise. Also long-term monitoring programs that allow us to create big zoological and botanical collections. There are human sources and facilities to conduct DNA analysis, including sequencing part at Institute Genetic and Cytology of the National Academy of Science of Belarus. Weaknesses are lack of direct financial support for project aimed at study of taxonomy and high price of sequencing. Threats are unpredictable economical and financial situations in Belarus and countries of CEE region, and future changes in the legal field associated with the new status of the digital sequence information in the framework of Nagoya Protocol. Anyway, we have good opportunities to get new data regarding invasive alien species, discover new species, strengthen our regional cooperation, and receive funding for a project through UNDP GEF program. That is all for now. Thank you for your attention.